Okay, we're going to continue with our second lesson in the chapter on biomolecules and biochemistry. In the first lesson, we gave an overview of biomolecules, and we spent a little more time talking about proteins. We talked about the different functions of proteins. They functioned as either being carriers or transporters, as enzymes, as structural molecules, defense molecules, hormones, etc. So we talked about their functions. We also previewed the fact that we would talk about other classes of biomolecules, polysaccharides, or what we can call carbohydrates, and lipids or fatty acids. We will get to those in other lessons. But in this lesson, we want to talk a little bit more about proteins. Remember that we said that one of the important concepts was that these important biomolecules, large biomolecules, macromolecules, are made from a relatively few number of smaller building blocks. And so let's focus our attention on the building blocks that comprise proteins. That is what we call amino acids. Now this slide may seem overwhelming, and don't worry, don't be freaked out about this. These are the structures of the 20 common amino acids. Now to a non-chemist, non-biochemist, non-biologist, these may look like a lot of little stick drawings, but they are very important molecules that have some common features, as well as being what we call the 20 common amino acids. We're going to zoom in on a few of these and show you what we mean. The amino acids all have the following part in common. They have what is called a carboxylate or a carboxylic acid functional group. That's that carbon with 2 oxygens. And they also each have an amino group. And the amino group and the carboxylate group are attached to the same carbon. That's why we call them alpha amino carboxylic acids. So that part is common to all 20. It's the same for all 20. Where they differ is in what is called their side chain. And the side chain is in the light blue in the ones that we've expanded. You may notice that one of these side chains looks a lot like benzene, an organic molecule which we talked about in our lessons on organic chemistry. Yes, that's the case. If I expand back out, the top row are all the amino acids which are considered to be non-polar. That is, their side chain does not like to interact with water. The next row are those amino acids whose side chain does like to interact with water, what we call polar amino acids. The different side chains give different chemical features to these amino acids. We also have a group which are called acidic amino acids, which has a side chain that has an acidic property like acetic acid or like vinegar. And we have a group that are basic amino acids. So the different side chains provide the different chemical features to the 20 amino acids. Now, the amino acids are linked together by what is called a peptide bond to form a polymer, which is a protein. This is much like the formation of polymers that we talked about earlier when we talked about, for example, forming nylon 66. This shows how that happens with amino acids. First, I show an example of 2 amino acids already linked together by what is called a peptide bond. And this drawing highlights in the dashed circle the peptide bond between one amino acid and another amino acid. And this also shows how a third amino acid can be joined together to the first 2 to form a growing polymer of amino acids. Now, when you string together these amino acids, you can form a long polypeptide, a polymer of amino acids. And this polymer can be 100 or more amino acids strung together. Now, in biological systems, the sequence of amino acids is not random. It has a particular order, and that order is determined by the genetic information in DNA. For example, a given protein, lysozyme, for example, will have a specific sequence of amino acids determined by the sequence of nucleotides in DNA. We may have covered already the genetic code, 3 bases code for a particular amino acid. Okay, now here's what is interesting. When you string together a sequence of amino acids to form a long polymer, this polymer has the ability to spontaneously fold up into a three-dimensional structure, shown in the bottom right side. And this is what is amazing to biochemists. What is actually happening is you're taking information that is in a linear sequence of nucleotides in DNA, translating it to a sequence of amino acids as a polymer of amino acids, a polypeptide or a protein. And then that protein can spontaneously assume a specific three-dimensional structure. A specific three-dimensional structure that can have things like active sites, if it's an enzyme, or it can have pockets that will specifically bind things like heme, if it's hemoglobin, or will have a particular three-dimensional structure that can recognize other proteins very specifically. The ability to form a specific three-dimensional structure is a key to the ability of proteins to function. Another example, immunoglobulins are able to recognize foreign molecules called antigen by binding to them very, very specifically. All of this is determined by the existence of a three-dimensional structure. Now, let's imagine that a mutation occurs or an error occurs in translating the DNA information, the genetic information in DNA, into the sequence of amino acids in the protein. There could be a couple of consequences. One is that the substitution or a mutation of one amino acid for another will prevent that polypeptide from folding up into a three-dimensional structure. That's one consequence. Or it may be able to fold up, but it may not have the amino acids in the right positions to correctly form the active site, to be an effective enzyme, or to recognize other molecules, or to be able to bind heme groups or other molecules that need bind to the protein in order for it to function. So mutations can lead to dysfunctional proteins, and in fact that's usually the case. We just sort of, from watching movies, we think that mutants are always more powerful. Actually, mutant proteins most of the time lack function. Let me summarize again what is really a very important concept. That is, genetic information is a sequence of nucleotides in what is called the genetic code. That sequence of nucleotides contains the information to lead to the synthesis of proteins, the sequence of amino acids in a protein. And proteins have the ability, most of the time, have the ability to spontaneously fold up into a functional three-dimensional structure. And then those proteins then carry out the functions such as those we described on the previous page. Important concepts. Now let me share with you something I think you'll find very interesting. These small building blocks have a feature which we call chirality, which is a complicated word, but it means that the molecules have handedness, that they're either right-handed or left-handed molecules. What does that mean? Well, if you look at the drawing to the left, I show two hands with molecules as if they were being held in the hands. First glance, you look at the molecules, they look identical. They've got one of those red COOH things, they got a carbon in gray in the middle, they got an NH2 thing at the bottom, a little bitty hydrogen on one side, and something called an R group that's sort of coming out towards you. So they have the same components. When we say that these are right-handed and left-handed molecules, what we mean is that they cannot be perfectly superimposed. They look similar, but they are like our left and right hands. If you ever tried to put on a pair of gloves and you get the right-handed glove and you try to put on your left hand, it doesn't work, and vice versa. The only time it works is if you're wearing mittens. But our left hand and right hands are not identical. They are what we call mirror images. Now, I'll pause and I'm going to do a little demonstration to try to explain what we mean by mirror images. Okay, here I am. I have my mirror and I have my left hand. This is my left hand. See, I've got my wedding ring. And if I bring, oops, it's hard to do. Oh, it's because I need to turn it the other way. And after playing around with it a while, you can see that in the mirror you see an image that will look like it is my, there's my pinky. It will look like it is my right hand, but actually the image is the image of my left hand just reflected in the mirror. And the giveaway, of course, is that I have my wedding ring, and that's on my left hand, but it looks like it's my right hand, just as if I bring my real right hand up, you can see that left hand and right hand, they are mirror images of each other. They may look like they're the same, but they are not superimposable. The thumbs will be on different sides if I try to stack them on top of each other. What a weird demonstration. Well, that was almost sad. It probably makes you wonder if I can get dressed in the morning. Nevertheless, the point is, molecules have handedness just like our right and left hand have handedness. This phenomenon is called chirality. Now, what is interesting is that all of the 20 common amino acids have the feature of being left-handed molecules. Not right-handed. All of them are left-handed molecules. And that means that all the amino acids that are used to make all the proteins that we have in all life forms, with just a few rare exceptions, are based on the L configuration. They are left-handed molecules. Now, in the bottom right, I show a few other molecules. I show something called L-glyceraldehyde and D-glyceraldehyde. Now again, this is chemistry, so don't get too freaked out about it. But L-glyceraldehyde and D-glyceraldehyde can be considered to be the parent molecules. That is, L-glyceraldehyde is a left-handed molecule. And we can consider all of the amino acids to be made from or derived from L-glyceraldehyde. Okay? D-glyceraldehyde, again, is a mirror image molecule of L-glyceraldehyde. They may look alike, but if you tried to exactly superimpose them by moving one over and trying to rotate it, you couldn't stack it exactly on the other, even though it has the same substituents, the same components. But what is interesting is that while L-glyceraldehyde can be considered the parent of all amino acids, and therefore all amino acids are L, D-glyceraldehyde can be considered to be the parent of all carbohydrates, from glucose and all other carbohydrates. And just as there exists no D amino acids, or they are very rare, all carbohydrates are based on the D configuration, with only very rare exceptions of there being L or left-handed molecules. Now these L and D stand for levorotatory and dextrorotatory. That means it rotates light, plain polarized light to the left or to the right. I won't burden you with that. But this is an interesting division of labor that exists in biology or in biological systems. Amino acids are all L, carbohydrates are all D. How about that? Okay, now of these 20 amino acids, some of them can also be described as essential. Now, by essential, we mean that we must have them in our diets. We must have them because we cannot synthesize them ourselves. About half, actually 9 out of the 20, of the amino acids are essential. We can make the others from scratch or from other molecules that we routinely have in our diet. But for 9 of the amino acids, we cannot synthesize them. We have to have them in our diet. Now, a consequence is that certain food sources are lower in certain amino acids than others. And this bar graph compares the availability of certain amino acids in eggs, hen eggs, and cornmeal, a dairy product and a grain. And it shows that in some cases, if we look for the amino acid leucine, we have about the same amount of leucine in eggs and in cornmeal. But if you look at other amino acids, one called isoleucine, one called lysine, one called tryptophan, the plant source, cornmeal, has a much lower content of these amino acids than does the dairy product. And this is something to be concerned about with respect to having a balanced diet, the fact that you need essential amino acids. Now, again, it's time that we pause and I'll ask you a few questions, and then we'll return with our third lesson in this chapter on biomolecules and biochemistry.